Good morning, everyone. Today we are going to cover the topic amoebiasis in this class. So, in this class, we are going to include what is amoeba, what is Entamoeba histolytica, amoebiasis as a health problem, habitat and transmission of this disease, nature of the disease, event of amoebiasis, host factor contribution, diagnosis, prevention, and treatment. Amoebiasis is a disease caused by a one-cell parasite called Entamoeba histolytica, although it is more common in people who lived in tropical areas with poor sanitary conditions. Amoeba is a single-celled protozoa that constantly changes its shape. Amoeba is a Greek word which means change. There are two types of amoeba. One is intestinal amoeba and the other one is free-living amoeba. Intestinal amoeba includes pathogenic and non-pathogenic amoeba. Pathogenic amoeba, which we call Entamoeba histolytica, and non-pathogenic amoeba, we call it Entamoeba dispar, Entamoeba moscovskii, Entamoeba coli, Entamoeba gingivalis. Free-living amoeba include Acanthamoeba species, Naegleria fowleri, and Bulmodium mandalis. Entamoeba histolytica was first described by Lumb in 1859 and Laws established its pathogenic nature in 1875 in a dysentery patient in St. Petersburg. Councilman and Leffer in 1981 described amoebic liver abscesses. Scorin in 1903 differentiated the pathogenic and non-pathogenic types of amoeba. Morphology of Entamoeba histolytica. Entamoeba histolytica has three stages. The first one is tropozoid. It is an invasive form of parasite passed only in feces of patients with active dysentery. Pre-cyst is the intermediate stage between tropozoid and cyst. Cyst is the infective form of parasite found in stool of the patient. Let's discuss amoebiasis as a health problem. Amoebiasis is estimated to cause 70,000 deaths per year worldwide. Symptoms can range from mild diarrhea to dysentery with blood and mucus in the stool. Entamoeba histolytica is usually a commensal organism. Severe amoebiasis infection occur in two major forms. Invasion of the intestinal lining causes amoebic dysentery or amoebic colitis. Now let's discuss the habitat and transmission of this disease. Entamoeba histolytica is present in mucosa and submucosa of large intestine. Amoebiasis is transmitted by fecal contamination of drinking water and foods, but also direct contact with dirty hand or objects as well as by sexual contact. Additionally, geophagy is a common route of infection in certain cultures. Nature of the disease. Symptoms are usually gastrointestinal, including diarrhea, vomiting, abdominal pain, or discomfort and fever. Symptoms take from a few days to a few weeks to develop and manifest themselves, but usually it is about 2 to 4 weeks. Most infected people are asymptomatic, but this disease has the potential to make the sufferer dangerously ill, especially if there is any suggestion of immunocompromise. Events on amoebiasis. The event starts when human being accidentally swallowed a cyst. When cyst enters the human body, it hatches in the small intestine and releases amoebas which divide into tropozoites. Tropozoites develop into cysts again, then exit the body in feces. Tropozoid again can invade other part of the body and reproduce by division. This is the picture which showing the same process of developing and multiplication of Entamoeba histolytica in body. Host factor contribution. Several factors that can contribute to influence the infection with Entamoeba histolytica. These factors include stress, malnutrition, alcoholism, corticosteroid therapy, immunodeficiency, alternation of bacterial flora inside the body. Risk factors include people in developing countries that have poor sanitary conditions, immigrants from developing countries, travelers to developing countries, people who live in institutions that have poor sanitary conditions, and HIV positive patients. The diagnosis of this disease can be done through different processes. The main and commonly used process is microscopy. This is the traditional means of diagnosis the disease. One simply look at the sample of stool under a microscope in this method. Next is serological diagnosis. This method includes the serological become reactive in invasive amoebiasis, indirect hemogglutination assays, ELISA, latex agglutination test, for-gel diffusion, countercurrent immunoelectrophoresis. Then the other one is culture. This is not routinely done because trying to get the amoeba to grow outside the body is very difficult and unreliable and is therefore not generally done. Treatment. Frequently, either metronidazole or tinidazole are used to treat amoebiasis. If this doesn't work, chloroquine, amethine, and dihydroamethine can be used. Eliminating cysts in carriers who do not have symptoms is accomplished with diloxanide furanate, idoquinolone, and paromomycin. Nitazoxanide is a newer drug that shows promise against not only Entamoeba histolytica but many other parasites as well. Prevention. Drink only bottled or boiled water which is stored in cans or bottles. Fountain drinks or any drinks with ice cubes are not safe. Water can be made safe by filtering through an absolute 1 micron or less filter and dissolving iodine tablets in the filtered water. Avoid fresh fruit or vegetables that were peeled by someone else. Avoid milk, cheese, or dairy products that may have been not pasteurized. Avoid anything sold by street vendors. Food safety and personal hygiene is very important to prevent this disease. Now, let's see if any vaccine available for this disease. Vaccines are being developed and tested for the treatment of amoebiasis. This vaccine is a modified version of the protein expressed on the surface of Entamoeba histolytica. A study in rodents found that the vaccine prevented the formation of liver abscesses, but much more research is needed to determine if these vaccines are useful and safe in humans. So, thank you so much for cooperating with this presentation and for your uh, interest. And I have included few questions to practice this topic with this presentation.